A very common question is, what is DNA? Well, DNA is a helical nucleic acid molecule that is capable of replicating and deciding the inherited structure of a cell's protein. But DNA is so much more than that. This is a DNA model. Model meaning a simplified version of something complex to explain or analyze it. Anyways, the DNA strand has two parts, the backbone and the middle part. The backbone is in a shape called a helix, giving the DNA the nickname double helix because it has two backbones. The backbone is made up of two parts, phosphate and deoxyribose, otherwise known as sugar. Phosphate looks like this, a yellow ball with other red balls around it, and the sugar looks like this, 6 gray circles and 1 red one. They connect to make the backbone of DNA. The middle part is made up of nucleotides. A nucleotide is necessary to DNA and is made up of a nitrogen base, a sugar, and a phosphate. There are 4 nitrogen bases, thymine, adenine, guanine, and cytosine. This is thymine. It's one of the 4 bases, connects with adenine, and is a pyrimidine base. This is adenine. It's a base, connects with thymine, and is a purine base. This is guanine. It's also a base, connects with cytosine, and is a purine base. This is cytosine. It's a base, connects with guanine, is a pyrimidine base. To get a better understanding of DNA, think of an egg. The shell is the phosphate and sugar, and the inside is the nucleotides. Each pair of bases creates what is called a gene. A gene is a unit of heredity information. Let's say you have blue eyes. The color of your eyes is a gene that you inherited from your parents. DNA strands and proteins make up what is called chromosomes. Chromosomes contain all genes and DNA strands, so they contain all heredity information. Chromosomes usually look like X's, and they're found in the cell's nucleus. All DNA strands can be cut. DNA is cut by restriction enzymes. Restriction enzymes are enzymes that cut DNA at specific points. This is a DNA sequence, and this is the restriction enzyme HAEIII. The enzyme will crawl along the DNA strand, reading the pairs as it goes along. Enzymes will continue to do this until they find the sequence they are programmed to cut. HAEIII cuts at the sequence GGCC. Once the enzyme finds the sequence it's programmed to cut, it cuts it, creating 2 strands. These new strands are called RFLPs. RFLPs stand for restriction, fragment, length, polymorphisms. RFLPs are DNA segments from restriction enzyme treatments. Gel electrophoresis is the separation of DNA strands according to size by measuring the rate of movement through an electrical field while in a gel. First, you put the DNA into the pipette, and then you put the DNA into the gel. You have to make sure that when using gel electrophoresis, the wells with the DNA inside are on the negative side. DNA is negative, so it will travel to the positive side because opposites attract. If the wells were put on the positive side, well, they would still travel to the positive side and end up in the buffer. So back to the question, what is DNA? The definition, helical nucleic acid molecule that is capable of replicating and deciding the inherited structure of a cell's protein, is right, but wrong. DNA is a complex tiny molecule that makes us who we are. It is the building block of life. Without it, we would be exactly the same, or nothing at all. DNA contains all of our genes and tells our cells what to do. DNA also determines our physical characteristics and what diseases we can get. DNA is the tiny, amazing little thing that makes us who we are. Thanks for watching!